Then when we talk about antimicrobial substances, we have complement system called complement. Why? Because it helps other immune cells to perform their function. They are basically the large group of serum protein which are produced in liver and help in destruction of pathogen. Complement do exist in an inactive form in the blood, and whenever there is a exposure to the pathogen, they become activated. They promote the inflammatory process, they enhance the all aspects of inflammation, and also cause lysis and killing of certain type of bacteria. So whenever a pathogen is entering into the body, there is activation of complement system. So if there is an antigen antibody reaction, it will lead to the activation of the classical pathway. But if there is no antigen antibody reaction, we have another pathway luckily, which is an alternative pathway where it is activated by the endotoxins which are present on the bacterial cell wall for polysaccharides, or maybe tumor cells, or even the zymosin which is present on the yeast cell. So this is alternative pathway. Then we have another pathway which is known as mannose binding lecithin pathway. The pathway is activated by unique type of sugar molecules which are present on the walls of certain bacteria. This mannose group in the bacteria cause the lecithin to bind with it, and finally it is activated. So no matter how a pathogen is being activated, be it classical, be it alternative, or be it lecithin pathway, the complement system gets activated. Certain facts related to the complement system. First, as you can see, the complements are represented by letter C, which is followed by a number. So here in this figure, you can see it is complement C, letter C, C1, C2, 3, then we have C4, 5, 6, 7, 8 and 9. Second, each complement is split into two, A and B. For 2 we have 2A, 2B, 3 we have 3A, 3B, and similarly it continues. A brings about the allergen response, whereas B is responsible for binding to the next series of enzyme resulting in a cascade activation. So A stands for allergen response. If it is C3A, it is bringing allergic response. C5A bringing an allergic response, whereas the B, that is C3B, binds to the next, C5B binds to the next enzyme, leading to the activation of cascade system. The classical pathway involved C1, C2 and C3. The alternative pathway begins with the activation from the C3 because no antigen antibody reaction is required. It is activated by endotoxins. Instead, it has two proteins which are important, that is factor B or factor 1 and factor D. Factor 1 is responsible for interaction with the polysaccharides in the cell membrane of invading cells, and propedrin of the factor D is responsible for stabilizing this complex. Here, look at this figure, the classical pathway being represented by red arrow and the alternative pathway by blue arrow. So they meet at a common point, and this common point is C3. So although they are complement is activated by three different pathways, they finally converge into one common pathway, that is C3, leading to the formation of C5, C6, C7, C8 and C9, which form membrane attack complex that disrupts the cell membrane and lead to cell lysis. Now this alternative pathway is also known as first line of defense, since there is no antigen antibody reaction. It is activated without antigen antibody reaction, and the binding of factor propedrin or factor 1 with the cell wall of the invading organism leads to the activation of C3 and C5, which can further lead to the activation of membrane attack complex. The classical pathway involves the activation of antigen-antibody complex. This complex C1 activates C2 and C3, and finally C3 will lead to the series of cascading events leading to inflammatory process and cell lysis. So the cell lysis can be done by one, opsonization, agglutination, then membrane attack complex leading to formation of pores within the cells, then chemotaxis and neutralization. The figure here depicts opsonization. The complement act as opsins. You can see the green colored ones which are coating the bacteria or the pathogen, so it appears more tasty for the phagocytes, so the uptake is facilitated. The process is opsonization. Here we can see the classical pathway, the lecithin pathway and the alternative pathway in a single slide. Classical is by antigen antibody complex activating the pathway. The mannose activating the pathway when lectin is binding with mannose, and then we have this pathogen which carries the polysaccharide which is activating the alternative pathway. All three pathways are then converging into this, that is activation of C3 into C3A by C3A convertase. C3A is then helping to mediate the inflammatory response, whereas the other product that is formed C3B result in opsonization. Then it is leading to the activation of C5 into C5A and C5B. C5B is binding along with C9, C6, 7, 8 and 9, causes holes to appear in cell membrane to the foreign body and form membrane attack complex resulting in cell lysis and activation, whereas C5A is responsible for mediating inflammatory response. C6, C7, 8 and 9 also promote the release of histamine and movement of macrophages toward the inflammation site. So this is whole of this represents the complement pathway, where the different products that have been formed are helping to enhance the inflammation and leading to destruction of bacteria, pathogen or any foreign substances that has entered. Let us try to understand the mechanism of formation of membrane attack complex or MAC after the activation of C3. It splits into C3A and C3B, and as we said, A is responsible for allergen responses, for example inflammation, whereas C3B will bind with C5 and causes it to split in C5A and C5B. Again, C5A will mediate inflammation, whereas C5B will bind with the complex that is C6, 7, 8 and 9. This complex forms the membrane attack complex and binds to the cell surface, resulting in the formation of pore. This pore will then causes the influx of fluid from the surrounding spaces into the cell, leading to the osmotic lysis of the cell or the cell death. This slide represents a summary of events that occur during complement activation. First is opsonization or the coating of a microbe to enhance the phagocytosis. Then we have phagocytosis as explained earlier. Then chemotaxis, also promoted by certain of the factors such as C5A and B, which causes chemotaxis. Then membrane attack complex, which is formed by C5 to C9. They cause the formation of perforin, and this perforin causes cell lysis. Then inflammation can be caused by factors such as 3A and 5A. Knowing the importance of complement system in our body regarding immunity, we will be able to appreciate that if there is a deficiency of any component of the complement system, it can lead to immunocompromise and overwhelming infection and sepsis. One such example is systemic lupus erythematosus. So what could happen if there is an uncontrolled activity by the complement system? It can be a serious condition because since it is promoting inflammation and in fact enhancing that, it can result in pathogenesis of various autoimmune disorder, and the reaction can spread just like a forest fire. It can be amplified and cause massive tissue destruction as such.